joining now. I think some people are still coming, but we can like keep adding them as we progress. This is on the Cassyni platform. At the end, a kind of edited version of this webinar will go online. People can ask questions in the chat. We'll have a discussion at the end, and this is a chance to ask, authors and Paloma from GBIF, anything you've ever wanted to know about kind of biodiversity informatics and GBIF and this process. And you can also ask questions for it will continue to ask questions for the next 24 hours as well. So the schedule will be I will do a brief introduction. We'll have two fantastic author talks, and then Paloma will close things, giving some GBIF perspective. And then there'll be time for questions. I can start sharing the. Ooh, we're getting some nice. Continuing to get lots of people to join, which is nice. So let me share screen. Hopefully this works, and you can see my screen. Is this hopefully working? Yeah, thank you everyone for attending. So we've done a couple of webinars before on data publishing, and so I won't go into so much detail on the background on that. I can share the links to the previous webinars. This one we thought we would get some more kind of author insight, and so I'm hoping to really we have two fantastic speakers, Catalina, Marceló-Díaz, and Paul Taconet, who will hopefully give some of this, insight from authors. Ooh, it's kind of a little tricky because I'm having to add people at the same time. But the fantastic thing about this series, this is now the third call for papers. It's been extended until June the 6th. But because this is the third call, we have the ability to show what's happened with previous papers, and the sort of lessons learned that we've had from doing this for about 2 years now. To give some just very for background for people who may not have come across this series before. Biodiversity data is crucial for so many things. We're in this climate change and a kind of biodiversity crisis in some places. Knowing this information is so crucial to address these issues. But this is a nice paper from about 10 years ago looking at species richness, predictions of where all of the biodiversity should be, and the sort of more tropical zones in the kind of the middle of the world. You can see richness is this kind of the top map is very kind of equatorial and in Latin America, Africa, Asia is where most of the species should be. But in terms of completeness and biodiversity records, so much of the map is blank, right? Gray, blue is kind of really missing data. So it's really crucial to fill these gaps. And talking about infectious diseases and vectors of human disease. Unfortunately, so many of these species are more prevalent and active in these areas where the biodiversity records are missing as well, right? You look at where malaria prevalence is, for example, and it overlaps in these sort of biodiversity sparse regions, which is a big problem. GBIF if people haven't come across it, is the home for biodiversity data. It's a network. It brings together many nodes and local instances, and there's over 3 billion occurrence records so far, and this number is very rapidly growing. Paloma will give us some overview on this at the end of the webinar. But our own personal part, I'm the chief editor of GigaByte Journal, and our own little part is one of these targeted calls for specific type of data, and in this case, thanks to very generous support from TBDR, the WHO, and great partnership with GBIF. We've really zoned in on one particular, this really crucial part of type of data, really trying to ensure that we and incentivize and make it as easy as possible to get this key data in. So as I mentioned, we've already done a webinar on what is a data paper. Just very briefly. A data paper is a peer-reviewed document describing the data set or groups of data sets. It takes obviously a lot of effort to manage, curate, get data together to share to a data repository such as GBIF. And so these data papers are a means of giving people credit, giving people recognition for that effort. But it also increases the visibility of this. It gives that kind of, the institutions, universities, CVs recognize peer-reviewed publications more than data. And so it's a way of kind of tagging onto that and getting credit for this hard effort, and adding all of this kind of additional contextual information that you may not get from reading the paper. So that's the rationale for data paper. Our GigaByte Journal is a particular home for this type of data, and yeah, we're doing a lot of work on getting this vectors of human disease data. So this is now the third call and third phase of submissions for this series. We've been doing it for a couple of years now. The criteria for consideration in the series is it has to be data on vectors of human disease. It needs to be shared into GBIF. We need a new data in this call. I think the last 2 years, as long as you've deposited it to GBIF in the last 2 years, it should be eligible. We want it reasonably, substantial number of records. Although there's flexibility with this, we understand that a lot of fields, this data is harder to get hold of. So we are flexible on that part. And the key thing is we want this data to be open. So needs to go under a CC0 designation. You can do that through GBIF. Thanks to very generous support from the WHO, they're covering the article processing charges, normally about $500 for up to 12 papers. The deadline is the 6th of June, and to make the data sharing side much easier, they have set up a help desk. Paloma will tell us about this at the end and can answer questions on the data submission side. But we're providing resources for publication costs, and also this additional data support. And our own GigaDB team also help and do data audits and data reviews on that side to make the process easier. So yeah, we'll talk more about this help desk at the end. But we're privileged now to have done 2 calls on this. We have about 20 papers in our series now, and that is a great opportunity to now look back and provide lessons learned. The fantastic thing that we've seen, I showed you the map and all of the kind of the missing data in the center of the world. From the first 20 odd papers so far, we've filled in some nice gaps. A lot of data in West Africa, Southeast Asia, Latin America, but there's still a lot of gaps to fill, and many different vector types. So the first two calls, we got a lot of mosquitoes, sandflies, ticks, rodents, kissing bugs, and snail data. Nearly 1 million specimens from 70 countries, many different data types. It's exciting to see not just observations, but specimen collections, imaging, citizen science projects, DNA barcoding. We've written a couple of papers summarizing all of this if you want to know more. Um, but the having done this for a couple of years now, it's really interesting to see the results of this. People may share data and not even know the how people are going to use this. In the last few weeks, a preprint has come out looking at the hosts of monkeypox, for example, the animal reservoirs for this, and linking to the far-footed rope squirrel. And so in the second call, we had a data paper on small mammals, data from West Africa, which included a lot of observations of this far-footed rope squirrel, right? So sharing data in this manner, you may not even know how important it is. Putting it out there, can have amazing consequences. And so this is why it's a great opportunity to now hear from a couple of the authors of the series, and talk through, and they can give their own insight on how the process was, and the downstream feedback. If you have papers, data sets of your own that you're thinking of submitting, then actually seeing that the end products and hearing from the authors is the best way to do this. So based on that, I will shut up now and let the speakers take over, because they're much more interesting than I am. So the first speaker, I think, will be is it Catalina first or Paul? It's Catalina
I'm
first.
sorry. Yeah.
So yeah, I will hand over to Catalina Marceló-Díaz, who will tell us about some of the work that she submitted to the series now. So Catalina, you should have access and should be able to share your screen.
Okay. Thank you, Scott. I'm going to try. Um, just let me see. Okay. Mhm. And maybe can you confirm if you can see the screen or not, or not yet?
It's still showing a Zoom. But your screen is sharing, but I don't see the slides. I think you need to. Okay, here we. Yeah.
Okay, just give me a
Slides
minute.
now.
Uh, I'm going to try to do. Oh, yeah. And now maybe it's better. Yeah. Okay. Thank you. So just give me a minute. I'm going to start. Okay. So hello, my name is Catalina. I'm from Colombia. I'm currently working at the National Institute of Health. But today we are going to talk about spatial patterns associated with the distribution of immature stage of Aedes aegypti in 3 dengue high-risk municipalities of southwestern Colombia. But more of that, I would like to explore how we made the data sharing insights about the produce and how we generate the data paper also. So can you see the screen? Yeah, yeah, it's okay. Okay, thank you. So let's start with a brief introduction to Aedes aegypti. So Aedes aegypti, as maybe of many of you know, is a key vector for dengue fever, and also is a very important mosquito for us because this mosquito is implicated in the transmission of different kind of virus, such as Zika, yellow fever, and dengue, as you know. We already have been working in a project about about to identify the principal containers and escape and these sorry, the all the containers, and where we can find this mosquito and its immature stages. Okay, so as you already know, dengue fever also affects approximately 400 million people annually. And in Colombia, we have Aedes aegypti being the primary vector, but also we have another vectors such as Aedes albopictus. Okay, uh, as just I'm doing all of these brief introduction to show you how uh, after this we could potentially improve the vector control strategies after we made of the data release. So in Colombia, we have different kind of potential containers where we can find the breeding sites for this mosquito. As you can see, an example of the main containers that we have. For example, we have here some barrels, some tanks, some little recipes, some bottles, tires. Also here we have plants. Also in Colombia, is very common. Sorry. To find this vector in the in the plant. How can I say, in the plant axils of bromeliads, and you can find some little containers with water inside of these plants, and sometimes this could be like difficult potential breeding sites to identify by the officers working at the Secretary of Health of these kind of institutions. And the importance of studying immature stage for us is that with the studying of this immature stage, such as larva and pupa, we can provide insights about the breeding habitats, also spatial information, and improve the information about the bionomy that we have, because sometimes we can find this species with another species, for example, Culex and Culex larva and Aedes larva usually come together in the same container, right? Following the methodology established by Romero in 2007, we made of this effort of sample, doing, not just for pupa, also for adults, but the results about the adults is also another published in another paper. And we found about 3,000, about, yeah, 3,100 pupas and larvae. And we report this using the tools provided by the GBIF. But I'm going to speak about this in a little bit. One insight very important for us about to do this kind of sample effort is to do the spatial distribution analysis to try to understand all this spatial information. To do this, we make them study in 3 municipalities of Cauca department, Patilla, Miranda, that is in the northern of Cauca, and Piamonte. And we make them sample the mosquito sampling in the municipalities, in the main municipalities of this region. And we established patterns using spatial information system tools. And also we found a very important thing, this is that, as you know, is common use very different kind of indices to surveillance in dengue. For example, we use larva indices, right, Breto indices, and others. But we know now that it's most important for us to make the surveillance with the pupa, right? Because pupa also has less mortality than larva. And we found in this paper and in others that are related with this paper that if you had some hotspots with a significant density of pupa, you are going to have a density very high with adults. If you have, if you found pupa spots, of course that you are going to find adults after that. But it was we found after make all the spatial analysis that it was, like consistent, that every time that we found a house with a very big, very big size of pupa there, instantly after the next time that we make the effort of sampling, we found the adult. And for us, and for all the region, we found that this was a very important result. So we make of this mapping effort of this mapping analysis. We try to, I remember to speak sometime with Scott that maybe it's try to difficult to find the limit when you are making the traditional paper and also you are making the data paper, right? So we try to to know you're going deeper with our results, and because we have another papers that you can find when we go deeper in these results. But finally, we published our paper with the spatial patterns associated with the distribution of this immature stage. And after that, what happened? It was very nice that we could show these geographic areas of interest to all the community, not just for the region, but that the impact that we can find in these results is very big, is very important for public health. Um, After that we published the paper, sorry, before that published the paper, we found very useful to make a good relation with the node in Colombia that is called SIB, Sistema de Información sobre Biodiversidad de Colombia. And I would like to show you this useful diagram. I know that it's in Spanish, but it's very nice because you can just, like make a summary about every step that you can find after make the, before that you go to the make the sampling, go to collect the data about your mosquitoes, and after finally found all your data with the visibility and all the parameters that that can they expect. Okay, so we start here, like make all the data quality, all the process of verification. After that, they SIP Colombia help us to make the integration and publication of by IPT tool. And after and finally, we made some process at the same time, such as make the data paper in this time with GigaByte, and also you can find another links or another tools related, such as the portal in SIP and also GBIF portal. But we found, I'm going just to share with you the two experiences that we already did in this data sharing. I put here just a little picture about a nice paper that I found that is using our data paper data. And I found this very nice because it's also in Colombia, but it's in another place that is very far in the south of Colombia. They are a group of researchers. I already speak with one of them, that is Rafael, and it's very nice that you see you don't see maybe the spread of your data sharing instantaneously, but you see this data sharing maybe 1, 2, 3 years after. But they sometimes, you found many things in the field that are not properly documented, right? So they use some of our data about the kind of containers, the kind of recipients that we can found in the field, because you know that in different kind of regions in Colombia are different. Yeah, sometimes we just say that okay, yeah, we have just these plastic containers of these things that you have in your house, but it's very different in different parts of the country. For example, right now we are make a project in La Guajira. That is a north area in our country, and we found another different kind of recipients that could be a risk for the people. So sometimes I found in science that you used to generally, I mean, you used to say something about things, right? But in different kind of geographic spaces are different. So, but this is not the only way that you use to data to sharing, because of course that all of us want that our papers are uh, would be known by the scientific community, but also it's very nice. Another way to make data sharing is about this very nice tool that also Scott promote us to use. There is the protocol way that you can, with a link, just put all your protocol and make this protocol enough clear and enough enough I don't know, concise maybe to know for the people. And in this data release, that it was the first data that we published, we used this protocol tool. And it was for us, it was at the first time, it was like, oh, it's quite strange. But after that, many of our colleagues and also students, um, they ask to have more information about this protocol. I mean, it's quite short, but sometimes in public health, you don't have you know, the updated tools to make the surveillance at the right time. So I found that this tool was very nice for us. And also, we have another benefit of data sharing. In this first data paper that was published, I guess that it was in the 2020, 21, and we already found many of these different kind of works that are using our data and are using our findings about this. But I want to result, or I mean to make a special mention with this, that is diversity analysis, distribution, and abundance of mosquito, because it starts like a thesis, right, made by some students, and after they don't contact us directly, but it was very nice that we can see first how made the thesis, and after finally in this recently in January they published the paper, and it was in the same territory as we made the study. I mean, in Cauca, that sometimes is not well known for all people, you know, because I know that it's difficult for us to maybe to see now in our minds where is located all of these places that I said. But I think that sometimes we think, or we want that our findings have a good or enough spread in all the world, I mean worldwide. But it's so important to spread the knowledge. I mean, local first, and after region, and after in across the country, across the region, and others. And okay, so just to sum up, I just want to show you some challenges and benefits that we found in all of this process. I I think that in every step that you have when you make a publication, you have different challenges that we need to solve, right? But in every step, also you have people or a team that is working with you. I mean, Scott, for example, in the final step with the publication, in the step two, your team, your work team, in the step one, the SIP Colombia, because in the first step, you need to have a data quality that could be enough good to make after that the publication, right? So second, maybe in the second step, I think that one of the challenges for all us, it was like the intellectual property, because I found a good thing in this kind of, metadata and that, I mean, not the data paper, but the data that you publish before in the GBIF, and is that you are make a recognition for all the people that is involved in the project, not just the researchers. I mean, you are make a good recognition about the stakeholders with the also with people that is that may help you to make the collect in the field. That is you can put in this metadata, and also in the database you can put who people is joined you or is joined the team to make the sample afford, or also who people is doing the identification. So I think that is good make the recognition of these people. And finally, in the step three, talking about the publication, sometimes it could be a challenge to accomplish the journal requirements and also the budget. In that way, I found that, for example, this call that is open now by GigaByte is a very good opportunity for people. I mean, for all the scientists to try to join the calling. For example, for us, it was like that. It was like we found the call and we found that this was related with GBIF. It was, a very nice process from the review after they published all the paper, and it was very nice to have a good team in the head of Scott, that make feel, like they give us the hand to explain the process. Because sometimes in some journals it's like, they just send you some emails and you need to do whatever you need to do. Okay, so I think that another benefits of data sharing is also that you can validate your findings. Also, we can see in another data papers, or also in another databases, another different kinds of sampling methods, and also kind of results. And we found, or we thought that ultimately we are going to contribute to improve the control strategies to not just dengue, I mean, to another vector disease. Also, you have future opportunities, and also some practical implications with these opportunities of research. For example, we have we feedback about data. I think that the main data users of vector-borne disease are the people that is in the territory, right? The people that works in health in the territory. So and again, I need to say that SIP is made a really nice job to try to promote all of these strategies, and also at the National Institute of Health, we're trying to improve the communication, to try to have more data papers in vector-borne disease to the community. But I think that that one of the most important reports that people in the territory say to us is that this data enhance the understanding about the distribution of Aedes aegypti, because sometimes public data in health quite, quite not open. So it's like a very complex situation, because it's public data and you need to have access to this data, but you have you found many barriers, one, another. But I think that this is a good start to try to change our mind and start to think that public health need to be linked to public data to make the decisions. And also another public health applications. I would like to just show you. Right now, we are working in a project in La Guajira that is in the north of the country. And before, we used to be the user that publish the data, make the data paper. But now we had like a challenge that it was like make the distribution of Aedes aegypti, Lutzomia, and Anopheles also in this department of our country that is in the northern of Colombia. And it was very difficult because we don't have data. Yeah, so just data. Just we found data very, around 1960, 1980, very old. It was the data that was published about this. But with GBIF, we made like three different sources. One of them, it was using data from GBIF. Another was using data from papers already published, but quite old. And the other data that we used was some samples that we did in the region, specifically in the Manaure municipality. And finally, we made this Maxen, this potential distribution model, using also some landscape shapes, and using not of course, the bioclimatic information that is available, but also we include another index like NDVI and EBI, and we made this. But it was very nice to when we have this challenge to think, okay, which what sorry, it was very nice to find that we can find the information in GBIF to do this, for example, right? Of course, that again, and I want to ask, and I want to speak one more time about the challenge is the data quality. Yeah, because you download all the information, all the databases, but after that you need to make again a data quality, and to be sure enough that you are going to have a good data in your map. But we are working in a paper about this, so hopefully I want we are going to share when it was finished. And also, as I said before, I think that all these kind of data sharing, specifically in the public health, helps to find future research opportunities, because you are going to find more collaboration with interdisciplinary teams. Yeah. And this can lead to innovative methods in vector control and surveillance. Yeah, I think that that, for example, now in Colombia is compulsory that you need to report your data collect in the SIP. Yeah, if you want to apply for one project, you need to report your data to SIP. So I think that if you put this or put this data sharing policy, yeah, in the way that you make science, you are going to you should do it. So people that maybe before in the past think that this was not necessary, now think that, okay, I need to do it and ask to SIP, ask to GBIF. So I think that is a good start, and we are in the group of entomology very proud of join all of these opportunities. So thank you. That's all, and sorry because I'm quite nervous, so I speak a little a little fast.
No, thank you, Catalina. That was amazing. That's exactly what I want to see. And thank you for your endorsement of what we've been doing, what GBIF and SIB have been doing. But that's very kind. But I'm most excited to see the examples of downstream reuse and the credit that you're getting for this, and that that's ultimately what I want to see. We want to see people using this in a public health context and tackling these diseases. So it's fantastic that you're tracking. You can see this from the data paper, and actually from GBIF, you'll be able to see this even more. Like this is probably the tip of the iceberg, and Paloma can say a bit more about this at the end. But it actually tracks all of the data that's not published yet. People using this data in kind of composite data citations and stuff. So but yeah, this is really fantastic. I'll keep the momentum going and hand over to Paul now because it will be great to see Paul's example now. Take it away, Paul.
Okay, can you see my screen? Yes, okay. So thank you, Scott, and thank you all for attending this meeting. So I'm Taconet. I'm a French researcher at the French National Research Institute for Sustainable Development in France, and I'm going to give you another overview of my experience with publishing the data here on malaria vectors in the context of the GBIF and GigaScience data call for data paper. So quickly, the context of the data that I have published. It was collected in the frame of a project in Côte d'Ivoire and Burkina Faso, so in West Africa. Project that aimed at assessing the impact of vector control of malaria vector control strategies of various strategies. So we had 50 villages here in Côte d'Ivoire and Burkina Faso, and during 2 years between 2016 and 2018, we collected entomological data on malaria plots. So those were our data. We had 8 mosquito collection rounds during 2 years in the 50 villages. At the end, we collected more than 60,000 malaria vectors. And maybe the specificity of our data was that it was fine-grained. I mean, we had fine-grained information at the individual mosquito scale. So we had date and geolocation of collection, but at the individual mosquito scale, we had the biting location, whether it's bite indoor or outdoor, priority states, infection to Plasmodium falciparum, and presence of genetic mutation linked to insecticide resistance. So we really had this kind of huge data set. So with the project team, we said we saw the call, and we said, okay, let's publish the data and write a data paper to for this data. So the first step was really formatting the data. First, the first thing I did actually was to read the very exhaustive documentation in the GBIF website. For all the people that need to publish the data, I can suggest the website. There's a lot of things already here if you want to publish your data. Okay, I understand how to publish occurrences and environmental data in the GBIF, but how to deal with the information at the individual mosquito scale. I couldn't really find the way to publish this data on the documentation. So I wrote an email at the help desk of the GBIF and GigaScience Journal, presenting my data and my problem. And 2 hours later, really fast, I received an answer from the help desk, and we exchanged some emails. And 1 week later, I had, let's say, my solution. So I knew how to publish and integrate the data, the individual scale, mosquito scale. Okay, so this is kind of technical, but in the end, I choose this down core schema to publish my data. In the event table, I had the dates and geographical coordinates of the mosquito collection sessions. In the occurrence table, I have the genus and the species of the collected Anopheles, 1 row for each mosquito collected. In the extension called extended measurement of fact, I have the information of the individual mosquito scale. So this extension fit actually my need to publish data at the mosquito scale. And lastly, in the measurement of fact extension, I had the environmental data of the sampling event time point. So once this was defined, I wrote some R scripts to come from my original database to the Darwin core schema. And lastly, no, not lastly, but second step was to publish the data in the GBIF. So I had my four CSV here that I presented before, and I used the IPT integrated publishing toolkit to publish the data in the GBIF. It's a quite intuitive tool actually when you know it. I had still some questions, and I could rely on my on the French GBIF endpoint, so the local really the local endpoint, to answer my question about the mapping between the data set, about some question about the metadata, the use of the tool, etc. I had a few email exchanges, and I could really like better understand how the tool works. So at the end of this process, I had my data set published in the GBIF with the URL. Last step was to write and publish the data paper. Was a actually quite easy and not time-consuming step. Once you have published your data on the GBIF, you already have all your metadata details, so it's not that long actually to turn it to data paper. It's the standard process for publication of data of articles. Sorry. What I really enjoyed is here these points, edits by the GigaScience editors. I mean, there was some editors in GigaScience that reread my paper and propose some modification to improve clarity and flow. That was really enjoyable, actually, especially for the non-native English speakers. And so at the end, the data paper was published. Feedback received. So the data set, actually in the GBIF, you have the number of citations. So the data set was already cited 19 times. There was some data on some article on biodiversity data sharing, on the impact of climate change or globalization on mosquito biology, etc. So various topics, reusing our data set. I know that the data set was also used by the GBIF directly for international training workshop. And since then, I have also published many other data sets in the GBIF. I've tried to convince my colleagues to publish the data, and it actually worked quite well. The most difficult is the first one. Summarizing quickly, for me and for us, it was really a nice way to have our data fair. In case of complex data, this is kind of more of an advice. If you have some complex data, take time to find the right formatting. There should be a format that meets your case and enables to integrate all the information. The GBIF format is quite adaptable, so you should find a way at the end to integrate rich information to have such information in the data. And do not hesitate to contact the GBIF helpdesk and the GigaScience editors. They are very supportive, as I tried to say in my presentation, both in terms of contents, for example, how to format your data, and from like technical assistance with the GBIF publication. And last thing that I didn't put here, we wrote a bunch of papers based on this data, so our team, and it's a very nice way to not always rewrite all the methods in the paper, like how we collected the data, etc. That's it. It was quick, but happy publishing.
Oh, thank you, Paul, again for the endorsement, but also, again, I'm excited to see that 16 people have there's at least 16 examples of reuse of your data, right? That's what we all want to see. Okay, so we are running out of time a little bit, but and it's late for people who are calling from further east, but we can maybe go an extra 5-10 minutes later, especially to answer questions. Paloma can now give a bit of insight on the from the GBIF perspective and also the working group, and then hopefully we can quickly answer a few questions. So we've already got one question in the chat, but if other people have questions, please add them. Okay, Paloma, take it away.
Thank you, Scott. And thank you, Paul and Catalina. It was very nice to see your talks. And I guess I'll be very brief because you've already mentioned a lot of the things I would talk. So I'm going to talk a little bit about GBIF initiative on open data on vectors. And it's not moving. Okay. Just a little bit about myself. So I'm a medical entomologist. I work at the Fundação Oswaldo Cruz in Brazil with sandflies and leishmaniasis. But since 2023, I've been working as a contractor for GBIF to support vector data mobilization. So I guess my talk is a little bit about publishing and about the work in GBIF. And when I was still a researcher at Fiocruz, I published a few papers in the first call. And so I guess just very briefly, the two highlights. One was the catalog of the sandfly collection that I was one of the curators. And the other paper was also work we did a long time ago, and it was sitting kind of in the drawer, and we didn't really have a good opportunity to publish. And with the call, we found the right place to make the data we collected in indigenous areas in the Amazon available. So it's for us was a good way to understand the life cycle of the data from the field to, even after some years, how to make them publish open and publish in a big repository for other people to use. So I guess you've seen the symbols for FAIR. One thing about GBIF and GigaByte is to make your data available as not only open, but also FAIR, which means that you can find on the internet, is accessible. So it has like a persistent identifier or URL, you can find it. And it's interoperable. So for biodiversity data, we use the Darwin Core standard, which is a standard that can handle quite well vector data. And The last thing is that the data is reusable. So as you saw from Paul and Catalina's examples. So all this is to make your data fair, it's good to increase the reproducibility of our work. This is something we talk a lot about in science, and to provide credit and acknowledgement. Also, Paul and Catalina mentioned that, and from my own experience, it's good to, you can acknowledge the work of field workers and technicians in a way that sometimes in papers we don't get the space to do. And also it makes the data quite visible. So it makes our work very visible to others. So it opens to collaborations. And so GBIF is the Global Biodiversity Information Facility. It's international networks and data infrastructure. So as of today, it's the largest repository for biodiversity data with, as Scott mentioned, over 3 billion records. And it's set to provide open data on biodiversity for everyone. So this is the network is not the most updated. So this is from, I guess, January. So it's the numbers, they change daily. But you can see the institutions that have published data in GBIF. Not all of these countries are members and of the network. In some areas like Africa and Asia, most of the support comes from GBIF's regional support teams rather than the national nodes. Yes, Colombia is a big publisher, a big user of data as well. And so this is to summarize GBIF's initiatives on health. So there is a running task group with experts advising on how to mobilize more data in health. There are a few publications that came out in the years since the task group was set up, which was 2020. We had a training workshop for improving data sharing skills that was set, and again, with the collaboration of the TDR. And Scott also shown the graph from the previous calls. And GBIF has a page, and I invite you to visit GBIF and Health with more details. And this is a bit of a summary of how the support. So as Paul and Catalina explained, we have our data. We need to map it to Darwin Core. So we need to structure our tables into an interoperable standard, which is the Darwin Core. And there is a lot of support from this technical support from the national nodes. So these are nodes that were helping with the publication of data sets in the 2 previous calls. So there's data from Argentina with the support of GBIF Argentina, in Colombia, in Brazil, in the Netherlands, in Belgium, and you can see all here. And for countries that do not have a node, they are not participants of GBIF, there are support teams and also help desk that can be reached. And so this is how a data set will look like in GBIF. And as you can see, there is affiliation, so the people responsible for the data, there is, so you can give attribution to everyone. And one nice thing is that once your data set is published in GBIF, it gets assigned a DOI, a digital object identifier, and GBIF tracks the citation of this DOI. So every time someone uses data and cites the DOI from your data set, it's GBIF will make it available in this citation link. And for us as researchers, it's nice to know, it's not only nice to know how people are using our data, but it's also important to help us show our funders and our directors and our bosses the impact of the data we produce. So this is a very, very nice feature. And we are seeing that more and more other metrics are used to give us funding or to assess our, our professional activities. So not only our papers, but also data sets and other outputs of our research can be used to, show what we're doing. And so finally, I guess I'll just repeat a little bit of what Paul and Catalina said, but important aspects of making our data available is the recognition of the work carried in the field. So a lot of us working with in medical entomology, we know how taxing and time confusing and money, it's labor, it's all the field work and then the lab work we do. And sometimes we don't just have the space to acknowledge everyone that is involved. And sometimes we have very big teams. And we also become more aware of the importance of producing good quality data. So data quality is something that, again, both Catalina and Paul mentioned. So we get more awareness of what it is to produce data that is complete, and it can be used by others. There is an increased visibility. So GBIF is, it's a very big and used repository. So we get a lot of exposure of, what we do. And there is contribution to the global knowledge of biodiversity. And so we help fill those gaps in the map that Scott showed in his presentation. And we get more opportunities to collaborate with others. And to me, again, the tracking of the data use, so tracking the citation is a very, very interesting feature that GBIF provides. From my own experience, we have used to show our directors that, for example, our collection is relevant. So I guess that's all I had to say. And I guess we open for questions now. Oh, sorry. Thank you very much.
Yeah, we've sort of technically run out of time, but we can carry on a little bit longer if people do have questions. Please put them in the chat, or I can even give you permission to speak if you like. We have one, so I see one in the chat from Anissa saying, please, so the question is, can the data be published as part of a manuscript later after it has been published as a data set in GigaByte? So this is a lot of the rationale for data papers, actually, that basically it just focuses on the data, right? And it's validating. And there was a little bit of discussion and mentioned previously on the data quality and how can we trust and stuff. So the beauty of the data paper is we're adding an extra level of peer review. Humans are at least kind of validating and scrutinizing this, so we can, this is another thing that data papers add, is there is this additional level of human validation beyond the excellent tools that GBIF have on automated validation. Like the IPT has a great kind of data validation tool. But anyway, this, the data paper is validated, independently validated data, and then we want people to use it so other people can do their analysis. But as the author yourself, then the rationale is you write your own analysis papers, your studies using and effectively validating the data further. And the publishers are all very happy with this. It's well understood. Most of the major publishers have data journals now, so this is part of their business model. They are very, very happy for you to do this. So that is the answer to Anissa is yes. I don't know if any of you have examples or are happy to say more on this yourselves. Are there any other questions? So following that, then I just Catalina and Paul, can you say anything about now your papers are published? Are you using this data in follow-up work?
Yeah, I mean, as I tried to tell at the end of my talk, the data that we published was part of a kind of a huge project with a lot of PhD students, with a lot of people. We're actually now in the process of submitting the paper at the Lancet with the results of the main epidemiological and entomological results of our project. And we keep on mentioning the reference of the data paper and the data in the GBIF. So, and as I try to tell, it's very, for us, it's time saving because we don't have each time in each paper, we don't have to rewrite the full methods, methodology, data collection methodology, etc. So yeah. In particular, in the frame of a huge project with a lot of paper that will be published, it's very useful.
Because otherwise, this kind of stuff would get lost or not covered, right? If you're writing a giant study, it takes ages for them to come out, and there's so many different parts, right? And the big medical journal that will publish this, they're not interested in that, like but this is really super important for the data papers, is that we're scrutinizing and validating, and that boring information is actually really useful for us, right? And also the downstream data users, right? They want to know, they will also want to know those technical details. But yeah, if they find it just as a little aside that's like lost in a giant, the supplemental parts of a giant medical paper, that, these things, that this information is otherwise lost, but it's useful. And Catalina had a fantastic example with the protocols as well, right? And I'm super happy. And I need to tell Lenny from Protocols.io about your experience, and I'll send him the video. Because he'll be happy to see that actually people are using the protocols on top of the data. So this is another great selling point for data papers.
Yeah, I want to just to be them. I'm agree with Paul, because if you take the good time to write the data paper, I mean the methodology, all the sections, is going to be. I mean, when you do this. Before they publish the data paper, you try to make your best to publish the data set, and after it's going to be easy to publish the data paper. We had a good experience with this data because we had a young researcher and student that she was joining in a internship, and we tried to teach her how to use this, how to 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 start in this knowledge of the Darwin core, the standard, and it was very useful for her, because after that she got her first publication, and she started her master's degrees. I mean, this year. So I think that because you have this kind of data paper in the different way that you use to work a traditional paper, is a good way to motivate the students. Because, for example, for some students, it's quite like, oh no, it's in a paper. No, I think it's like very difficult to start to writing, and you need time to writing. You need, it's quite. It's a challenge, but a data paper could be a good start for an student, because it's so clear, the temporal scope, the geographic scope, all is so clear, and after that it's going to be easy to make the jump to make all the data paper. And I have a question for Paul, maybe because I found his lecture very interesting. He explained about also that he take some landscape and data climate, maybe, but I'm not sure. You when we make the publication of this data set, Paul, you put all this data about climate and about landscape that you show us in GigaByte repository, or also in another platforms similar, like GBIF, but for, I don't know, for climate or something like that. Just I have that question.
So if I publish my data on another, like the environmental data on another tool, on another repository? No, I only published it on the I only published it on the GBIF, yeah.
In GBIF, in GBIF also.
Yeah, as an extension, yeah.
Very nice. I didn't know that. I mean, I just thought that biodiversity, but not the other. Very
No,
nice.
that's really the nice point of the GBIF publication is that you can publish the occurrence, of course, the event, the occurrence, but you can also publish many other information associated with the data. So environmental data, but also really fine scale data, DNA data, and many other. Yeah.
We are at GigaScience. We have our own data curation team. A few of them are on this call as well. Hi guys. They're there to help for a lot of these other if there was other supplemental data. We've had a few studies with big imaging. In like where it's supported by images. So we we can host the images and some of this other supplemental kind of data types as well. And that's another thing that we add is we provide this additional assistance. Yeah. I think we've kind of run out of time now. I don't know if Paloma has any final comments. Otherwise, thank you for everybody for attending. I would like to thank particularly the speakers, Catalina and Paul. It's been super interesting. I'm just really happy to see we publish something, it's out there, but like to actually follow on and see what has happened to your kind of publication babies and the responses from that. It's super useful. This video will be it'll take a little while to edit it and then it will go live and people can refer back to it. There may be additional questions. And yeah, just a final plug for the third call of the series. The deadline is 6th of June. Please contact us, myself, the health at gbif.org, and Paloma will probably be one of the people replying to you if you have questions on the data and on the submissions. Yeah, we're super happy to help you get these things out. As you've seen, they're not that difficult to write, but can potentially have a big downstream legacy and response. So thank you everybody, and I hope to see lots more very fascinating and useful data. Thank you.